For 70 years, medical textbooks, they confidently told us that the human bladder was this perfectly sterile environment, like a pristine, germ-free room inside your body.
Right. Yeah.
And we literally built our entire modern medical approach to urinary tract infections around this one, supposedly unshakable fact.
We really did. I mean, it was foundational.
But when you actually dive into the modern research, you find out that virtually everything we thought we knew about UTIs, from that sterile bladder concept all the way to like drinking cranberry juice, it has all been completely overturned.
It's just a remarkable story of how science self-corrects, honestly. Because we operated for decades on these very fixed, almost plumbing-based assumptions about the urinary tract.
Like it's just a set of pipes.
Exactly. But when researchers finally got the tools to look closer, they found an environment that is incredibly dynamic. I mean, it's a highly complex microscopic ecosystem,
Yeah.
and understanding it is actually crucial to fighting one of the biggest public health threats of our time.
And that is exactly our mission for you today. We are taking a deep dive into the microscopic battleground of the urinary tract.
Yeah, it's a fascinating area.
We want to understand how these specific bacteria manage to invade, how they pull off this wild Trojan horse maneuver to hide inside your own cells, and why your immune system's attempt to save you is actually the thing causing all that agonizing pain.
The friendly fire aspect, yeah.
Exactly. Okay, let's unpack this. And I want to start with the biggest paradigm shift of all. The idea that a healthy bladder is a sterile, clean room. How did we get that so wrong for so long?
Well, it all goes back to the mid-20th century, specifically to an infectious disease physician in Boston named Edward W. Cass.
Okay, Cass.
Yeah, and in 1956, Cass was trying to solve a very serious, very practical problem in clinics. When doctors ask a patient to provide a clean-cut urine sample in a cup, that sample inevitably picks up stray bacteria from the surrounding skin, as it leaves the body.
Right, because the skin obviously isn't sterile.
Exactly. So Case needed a mathematical way to look at a petri dish and distinguish a genuinely dangerous kidney infection from a sample that just had a little bit of, skin contamination.
That makes sense.
And this was especially vital for protecting pregnant women, where undetected infections can actually lead to premature labor.
So he basically had to draw a line in the sand. He had to figure out at what point a few stray microbes become an actual medical emergency.
He did. And he proposed a very specific threshold. It was 100,000 colony-forming units of bacteria per milliliter of urine.
100,000. Okay.
Right. If the petri dish grew 100,000 units, you had what he called significant bacteriuria, and, well, you got treated. If you were under that line, you were considered fine.
It's like he set up a nightclub bouncer for the bladder.
A bouncer? I like that.
Yeah, like the bouncer's just standing at the door with a clicker, and if 99,000 bacteria show up, he shrugs and says, it's a quiet night, go home. But the second it hits 100,000, he yells, riot. We have a riot.
That's pretty much exactly how we treated
And
it.
it just seems incredibly arbitrary, though, to apply one fixed number to every single human body.
This raises an important question, which is exactly what clinicians started asking when they realized Cass's number just wasn't working for everyone.
Really? Because it was a gold standard, wasn't it?
It was absolute gospel for decades. But doctors and clinics were constantly seeing women coming in with classic, totally miserable UTI symptoms, the intense burning, the urgent need to go.
Right.
But when their lab results came back, the colony counts were only like 100 or maybe 1,000. Under the old rules, doctors essentially had to look at a patient in severe agony and say, well, the math says you don't have an infection.
Wow. That must have been incredibly frustrating for the patients.
Oh, absolutely. Which is why modern guidelines, like the ones updated in 2019 by the Infectious Diseases Society of America, have finally dropped that rigid number.
Oh, they did?
Yeah. They realized that in symptomatic women, a much lower bacterial count is actually incredibly significant. Context matters way more than an arbitrary threshold.
Okay, that is a huge relief for patients, but it still doesn't explain the whole sterile bladder myth. Because if the old definition of a UTI was just bacteria where bacteria shouldn't be, when did we realize that bacteria actually should be there?
That realization came much more recently, actually, between 2011 and 2014. And it happened because our technology fundamentally changed.
What do you mean?
Well, for a century, we relied on taking a fluid sample and trying to grow the bacteria on, nutrient jelly in a petri dish. But many of the bacteria that naturally live in the urinary tract are incredibly slow growing, or they die the second they're exposed to oxygen in a lab.
Oh, I see.
So the dishes always looked empty, and we just assumed the bladder was totally sterile.
Right, because we just couldn't see them. So what changed around 2011?
Researchers started using a technique called 16S RNA gene sequencing.
Okay, that sounds intense.
It's basically a genetic scan. Instead of trying to grow live bacteria, this technology scans the urine for the unique genetic fingerprints of bacteria.
So they can't hide.
Exactly. And when they ran this highly sensitive scan on completely healthy people with absolutely no symptoms, they found a thriving, diverse community of microbes. They proved conclusively that the human bladder has its own resident urinary microbiome.
Which completely scrambles the definition of the disease. I
Yeah.
mean, if the bladder naturally has a microbiome, if it's supposed to have bacteria living in it, then just having bacteria in your urine is totally normal.
It is.
But having an actual UTI is something completely different, right?
That distinction is the cornerstone of modern urology. Just having bacteria in the urine is called bacteriuria, and it is often completely harmless.
Okay.
A true urinary tract infection is a disease state. It requires symptomatic tissue invasion and an inflammatory response that, overwhelms your host's defenses.
Got it. Okay, so if the bacteria are supposed to be there, how do the dangerous ones get in? Because I know female anatomy provides a shorter, steeper water slide, so to speak. The female urethra is, what, about 4 centimeters compared to the male's 15 or 20?
That's right. And is much closer to the GI tract. That sheer mundane geometry is why women get UTIs roughly 30 times more often than men.
30 times? Wow, so that explains the geography. But urine is constantly flushing down that water slide. How on earth do these bacteria actually climb up it against the current? Who exactly are the invading soldiers in this microscopic war?
The primary antagonist in this story is a specific type of bacteria that causes between 75 and 85% of all uncomplicated community-acquired UTIs.
Okay, what is
It's called uropathogenic E. coli, or UPEC for short.
Wait, E. coli, like the bacteria that already lives in our gut?
Yes, these aren't exotic pathogens that you catch from some strange external source.
Mm-hmm.
E. coli lives peacefully in essentially everyone's colon, helping you digest food.
Right.
But the UPEC strains are different. They are essentially heavily armed versions of your own gut flora.
How does a peaceful gut bacteria suddenly become heavily armed? That sounds crazy.
What's fascinating here is how bacterial genetics operate. Bacteria can actually trade and acquire large chunks of DNA from each other.
Oh, wow.
Yeah, these dangerous UPEC strains have acquired extra genetic cassettes, known as pathogenicity islands. You can think of them as specialized biological toolkits, basically extra equipment that ordinary gut E. coli simply don't have and don't need.
I am morbidly curious. What kind of tools are we talking about? Like, how do they get up the slide?
Well, that downward flow is their very first challenge. To keep from being flushed out, they deploy adhesins.
Adhesins, meaning they adhere to things.
Exactly. These are long hair-like appendages that extend out from the bacteria called pili. The most extensively studied ones are type 1 pili. And at the very tip of these structures is a specialized protein called VIM.
FIMM. And what does FIMS do?
FIM is designed to bind with just extraordinary precision to specific sugar molecules called mannose receptors, specifically uroplakins, that line the surface of your bladder cells.
Oh, wow. So they're literally deploying microscopic grappling hooks to lock onto the bladder wall.
A molecular grappling hook is a perfect analogy, and they have different hooks for different environments.
Oh.
Yeah, if they want to climb even higher, up into the kidneys, some strains deploy a different appendage called P-pili. These bind to completely different receptors found higher up in the tract called gal-alpha-1,4-gal receptors.
That's terrifyingly specific.
It is, and it's why strains with P-pili are disproportionately responsible for severe kidney infections.
Okay, that is super efficient. But hanging onto the wall is one thing. They still have to survive and replicate, right? What do they even eat in there?
That is their second major challenge. Urine is an incredibly harsh, nutrient-poor environment, and more importantly, it is highly deficient in iron.
Why iron?
Iron is absolutely essential for bacteria to metabolize and replicate.
Yeah.
The human body knows this, so it actively hides its iron away to starve potential invaders.
So how do the grappling hook bacteria get around the iron shortage?
They use another tool from their genetic toolkit, molecules called cytophores. These include compounds with names like aerobactin and yersinia-bactin.
Okay, cytophores.
Right. The bacteria secrete these molecules into the surrounding environment, and they act like aggressive chemical sponges. They literally rip the hidden iron away from the host's own proteins and carry it back to the bacteria.
That is wild. It's just a ruthless evolutionary race for scarce resources.
Exactly. Researchers have studied this heavily in a lab model called the UTI-89 strain. It's a very famous UPEC strain used to observe all these mechanisms.
So we have our own gut bacteria carrying iron-stealing backpacks and using Fimex grappling hooks to scale the urethra and lock onto the bladder.
Yep, that's the picture.
And for decades, scientists thought that was the end of the story, right? That the bacteria just held onto the outside of the tissue like barnacles on a ship.
That was the assumption, yes.
But how did that understanding change?
It changed thanks to a monumental discovery by Scott Holtgren's laboratory at Washington University in St. Louis.
Okay, what did they find?
Starting around 1998, they were using highly advanced imaging on mouse models to observe how these bacteria behave in real time. And they realized that the FimH grappling hook doesn't just hold the bacteria in place.
It does something else.
The binding action actually sends a signal to the human bladder cell, forcing the cell to swallow the bacteria whole.
Wait, here's where it gets really interesting. You're saying the bacteria are intentionally triggering their own kidnapping.
Exactly. The specialized cells lining the bladder, called umbrella cells, basically fold their membranes around the bacteria and pull them inside, trapping them in a little cellular bubble called a vesicle.
But instead of being digested, it's like a sci-fi movie. The bacteria break out of that little vesicle, enter the main body of the human cell, and just start rapidly cloning themselves.
They replicate at an astonishing rate.
Yeah.
They pack themselves so tightly into the human cell that they form dense, highly organized structures called intracellular bacterial communities, or IBCs.
IBCs.
Yeah, you can have tens of thousands of bacteria perfectly shielded inside a single host cell.
Which completely shifts how we have to think about treating this. Because if I take an antibiotic, that drug circulates through my bloodstream and filters down into my urine. It's going to wipe out any bacteria floating freely in the tract.
Right, the planktonic bacteria.
Yeah. But if the bacteria are hiding inside these microscopic cellular bunkers?
The antibiotic airstrike happens, but it cannot penetrate the bunker. The drugs just struggle to reach therapeutic levels inside the human umbrella cells.
Wow.
Furthermore, the bacteria inside these pods often shift into a dormant, slow-growing state. And most antibiotics only work on bacteria that are actively dividing.
So let me get this straight. As a patient, I take my course of antibiotics, I stop feeling sick, I give a follow-up urine sample, the lab grows it on a dish, and it comes back clean.
Yep, completely clean culture.
And my doctor tells me I'm cured. But a month or two later, the nightmare starts all over again.
This specific cellular mechanism explains one of the most frustrating clinical realities of this disease. The bladder's natural defense against infected tissue is to shed it, much like shedding dead skin.
Okay.
When that infected umbrella cell naturally sloughs off into the urine, it eventually degrades and bursts open.
Releasing tens of thousands of rested, surviving bacteria right back into the bladder.
Precisely. This is why 25 to 50% of women who suffer a UTI will experience a painful recurrence within 6 to 12 months.
Yeah.
The infection didn't come from a new exposure. The original invaders were just hiding, waiting out the antibiotics.
That is just incredibly devious. But wait, if the bacteria are safely hiding in these bunkers, just quietly stealing a little bit of iron to survive, what is actually causing the patient so much agony?
That's a great question.
Because I always assumed the burning sensation meant the bacteria were releasing acid or, I don't know, literally biting into the tissue.
It is entirely counterintuitive, but the bacteria are not causing the pain directly. The physical misery you feel is actually a product of your own body.
Friendly fire.
Yes. If we connect this to the bigger picture, it is a fascinating demonstration of how the innate immune system works. The cells lining your urinary tract have a built-in alarm system. They are studded with pattern recognition receptors, and the crucial one here is called toll-like receptor 4, or TLR4.
And what exactly triggers the TLR4 alarm?
TLR4 is designed to constantly scan for a specific molecule called lipopolysaccharide, or LPS. LPS is a major structural component of the outer membrane of E. coli.
Okay, so it sees the LPS, then what?
When the receptor detects LPS, the alarm trips. The bladder cell immediately begins manufacturing and releasing chemical messengers called cytokines. The key player here is interleukin-8.
So interleukin-8 is like the distress signal.
Think of it as a biological flare gun. It gets released into the surrounding tissue and creates a chemical gradient, a breadcrumb trail that alerts the heavy cavalry of the immune system, the neutrophils.
Neutrophils, those are the white blood cells, right?
Yes, very aggressive white blood cells circulating in your bloodstream. When they sense that IL-8 trail, they literally squeeze out of your blood vessels, migrate through the bladder tissue, and pour into the urine to hunt the bacteria.
And that massive influx of white blood cells is what doctors look for when they test your urine, right? A condition called pyuria.
Yes, pyuria is the clinical hallmark of an active immune response. But this battle comes at a steep cost.
The burning.
Exactly. When all those thousands of neutrophils rush into the tissue, they release their own inflammatory compounds. This massive inflammatory cascade heavily sensitizes the delicate nerve endings in the bladder wall.
I see.
That intense localized inflammation is what translates into the physical burning and the frantic urgency you actually feel.
And if that inflammatory war zone isn't contained in the bladder, the stakes get dangerously high, don't they?
Very much so. If the bacteria manage to use those P. pili grappling hooks to climb the ureters into the kidneys, you develop pyelonephritis.
Which sounds bad.
It brings high fevers, severe back pain, and a very real risk of permanent kidney scarring. In the worst cases, the bacteria break into the bloodstream, causing urosepsis, which can be fatal. And this upward spread is especially dangerous for young children who might be born with a structural defect called vesicular reflux, or VUR.
VUR. What does that do?
It acts like a broken valve, allowing urine to flow backward from the bladder toward the kidney, essentially giving the bacteria a free ride directly to vital organs.
That is terrifying. It's just wild to realize that the burning and the pain is essentially our own body aggressively reacting to the invasion.
It really is friendly fire.
But if the immune system's massive response is what causes the symptoms, how on earth are we supposed to manage it? Let's talk about the giant ruby red elephant in the room.
I think I know where this is going.
If someone feels that burning start, the immediate cultural reflex is to drink a gallon of cranberry juice.
The most famous folk remedy of all.
I really need to know, does chugging tart, sugary juice do anything at all to stop those molecular grappling hooks?
Well, the science here is a brilliant example of why laboratory theory doesn't always translate to patient reality. The idea behind cranberry juice is actually highly scientifically plausible. Yeah. Cranberries contain specific compounds called proanthocyanidins. And if you put those compounds into a petri dish with UPEC bacteria, they do, in fact, bind to and block that FimH grappling hook.
Wait, so it works in the lab?
It works beautifully in isolated cells. But when researchers conduct massive systematic reviews, looking at thousands of real human patients over several years, they consistently find that drinking cranberry juice or taking cranberry supplements does not offer a strong or reliable clinical benefit for preventing recurrent UTIs.
You mean millions of us have been chugging this tart juice based on a mechanism that completely fails in clinical trials?
Unfortunately, yes. The human digestive system, the concentration of the compounds, the timing, the real-world variables just overwhelm the petri dish mechanism.
Man, the cranberry juice empire is going to be upset. What about the other myths surrounding diagnoses? I've heard people say they know they have an infection just because their urine is cloudy or smells incredibly strong.
That is a very dangerous clinical myth. The clarity and odor of urine are dictated heavily by what you eat, how much water you drink, and what vitamins you take.
So it's not a reliable sign?
Not at all. Clinical guidelines emphasize that doctors should never rely on smell or cloudiness to diagnose an infection.
Oh.
You must look at the patient's actual symptoms paired with a proper laboratory urinalysis.
But even inside modern hospitals, doctors have been getting the diagnosis wrong because of the whole asymptomatic bacteriuria debate, right?
Yes, that has been a major issue.
Because for years, the conventional wisdom was that if a lab test showed bacteria in the urine, you hit the patient with antibiotics immediately, regardless of how they felt.
And that reflex has caused immense systemic harm. Unless a patient is pregnant, where the risks of kidney infection are just too high to ignore, or they are about to undergo a urologic surgery that will physically cut into the tract, you should absolutely not treat bacteria if the patient has no symptoms.
Really, just leave them alone.
Massive clinical trials have proven that treating asymptomatic bacteriuria doesn't prevent future infections. In fact, it does the exact opposite.
Wait, how could taking an antibiotic make you more likely to get a real infection later?
It comes down to the concept of ecological space. If you have a relatively harmless, lazy strain of bacteria living in your bladder microbiome, it is physically occupying space. It is consuming the limited iron and nutrients.
Okay, I follow.
If a doctor blasts your bladder with broad-spectrum antibiotics to clear a positive lab test, they wipe out that harmless resident strain. They essentially pave a massive, sterile parking lot, leaving it wide open for a highly virulent, dangerous UPEC strain to move in and take over.
We are intentionally clearing the way for the worst possible invaders just because a piece of testing paper turned pink.
It's a huge problem.
And this overtreatment happens all the time with vulnerable patients, right?
Oh, overdiagnosis is an epidemic in itself, particularly for elderly patients and those with urinary catheters.
Why catheters specifically?
Catheters are uniquely problematic because they bypass the body's physical defenses and provide a perfect plastic surface for bacteria to build biofilms.
What are biofilms?
Biofilms are thick, slimy, protective matrices that shield entire communities of bacteria from both the immune system and drugs. When a catheterized patient gets tested, the lab will almost always find bacteria because of these biofilms.
But they might not be symptomatic.
Exactly.
Yeah.
Treating them with antibiotics when they have no fever or pain does nothing but drive drug resistance.
Which brings us to the ultimate stakes of this entire deep dive. UTIs are so incredibly common globally that treating them is one of the single largest drivers of outpatient antibiotic prescriptions on Earth.
And this scorched earth approach of trying to sterilize the tract is actively fueling the rise of ESBL-producing E. coli.
ESBL.
It stands for extended spectrum beta-lactam. These are bacterial enzymes that have evolved to completely destroy our standard first-line antibiotics like penicillins and cephalosporins.
Oh.
By overprescribing antibiotics for asymptomatic patients or trying to cure every positive lab test, we have inadvertently bred superbugs. It is a slow-moving public health crisis.
Which is a heavy but, incredibly vital reality to understand. So let's look at the journey we've been on for this deep dive.
It's been a lot of ground to cover.
It really has. We started by tearing down Cass's 100,000 threshold and debunking the myth of the perfectly sterile bladder, realizing we have a natural urinary microbiome.
Right.
We looked at the sheer mechanical geometry of the tract. We saw how those rogue UPEC strains deploy FIM-H grappling hooks to climb against the current and use siderophores to aggressively steal iron.
The biological toolkit.
Yeah. And then we unpacked that terrifying Trojan horse maneuver where bacteria force our cells to swallow them so they can build impenetrable IBC pods to outlast our drugs.
Shielded from the airstrike.
Exactly. And finally, we realized that the agony of the disease is actually friendly fire from our own immune system's TLR4 alarm, triggering a massive white blood cell invasion.
It represents a total paradigm shift. We can no longer view the bladder as simple plumbing. It is a highly complex, microscopic ecosystem.
Like a fragile one at that.
Truly.
Yeah.
And because the threat of antimicrobial resistance is escalating so quickly, the future of UTI treatment cannot rely solely on inventing stronger antibiotics.
So what do we do?
The scientific community is aggressively shifting toward non-antibiotic strategies. Researchers are currently developing synthetic compounds designed to specifically block those FIM-H grappling hooks, preventing the bacteria from ever getting a grip in the first place.
That's amazing.
They're also promising clinical trials for vaccines that target the UPEC adhesins directly, training the immune system to clear the invaders without collateral damage.
We're finally learning to target the specific weapons instead of just carpet bombing the entire tract. So what does this all mean for the future of how we look at the human body?
If we truly embrace the concept of ecological space, the fact that harmless bacteria can physically block dangerous ones from taking hold, it completely changes our medical philosophy. Well, I'll leave you with this thought to mull over. If modern science has proven that blindly blasting the bladder with antibiotics clears space for the absolute worst superbugs to take over, could the future of UTI prevention look less like maintaining a sterile war zone and more like cultivating a garden?
Cultivating a garden.
Yeah. Could we, in the near future, intentionally seed the urinary microbiome with highly competitive, completely harmless strains of bacteria, like a customized bladder probiotic, to simply crowd out the invaders before they ever get a chance to deploy their grappling hooks?
Just take up all the space so the bad guys can't get in.
Exactly. It suggests that in the long run, the best defense isn't a stronger weapon. It's a healthier, more balanced ecosystem.
That is a phenomenal way to look at it. Thank you for taking this deep dive with us. Catch you next time.